All right, good morning. Today we are talking with Dr. Nicole Ford. Dr. Ford is an assistant professor in biosciences here at KCU. Dr. Ford got her PhD in molecular biology from Princeton University. And next she went on to do some postdoctoral work at the Medical College of Wisconsin and then the Pacific Northwest National Labs in Washington State. Dr. Ford then held a faculty position at Chowan University in North Carolina. I think I said that right. And she joined us here at KCU in 2019, right before the COVID-19 pandemic hit. So the lab has been kind of shut down for a while, but we're all in that same boat. So welcome, Dr. Ford.
Hi.
So what I'd like for you to do first, if you don't mind, is just give us a short elevator pitch about what kind of research you're interested in right now and what some of the projects that you're working on are here at KCU.
Sure. So right now I have one project that is active in my lab, and all of my projects really are more basic science and a lot of problem solving, trying to figure out what's going on at a molecular level. And the project right now that I'm working on has to do with chemicals that interact with our DNA. And what are the rules for where they interact in that DNA? And if we can figure out what sequences in the genome, I don't want to say attract, that gives them a little bit more than they have, but where they are most productive in their interactions, this is where I'm interested in finding those sequences. And if we can find those sequences, then we can figure out what genes in the genome are going to be targeted by these chemicals and these drugs. And if we can figure out what happens when we mutate those genes, then we can potentially find some of the side effects for these drugs or where some of the bad effects of environmental chemicals come from.
Okay, thank you. So that's interesting. When you talk about the chemicals that interact with our DNA, you mentioned environmental chemicals. What are some examples of, or an example of those chemicals that you focus on in your lab?
Yeah, so all of these chemicals are very flat. They're very planar. So they can slide in between the rungs of that DNA ladder and then interact with the nucleotides. So there are chemicals like this in cigarette smoke and automobile exhaust. Several of the cancer chemotherapeutics act in this way as well. I am interested in particular at this time in a drug called Sorlin, which is used, it actually has been used medically for thousands of years, not hundreds, thousands of years. And this particular drug, it's commonly used in overgrowth conditions, particularly dermatological overgrowth conditions. There are one or two cancers that it's approved to treat as well. But the idea is with all of these drug aspects, like I said, there are also some other cancer therapeutics, is that if you put enough of them into a cell, they're going to interact with the DNA, cause damage. If you damage the DNA enough, the cell is going to die. And when you're producing too many cells, that's the goal, to curb that growth. So at that level, at that therapeutic level, you're trying to kill them, not damage them and let them live. But not all the cells are going to get the therapeutic dose. So for the ones that do survive and perpetuate, what are the effects of the mutations that are caused by erroneously repairing these damages?
Okay, so the Sorlin chemical that you mentioned, this is part of or a component of pharmaceuticals, or is this in food, or where is this coming from?
So originally, yes, you can find it in several foods. There are specific foods that are high in sorlins. However, at this point, it's more of a purified molecule, specifically usually in a cream. like I said, dermatological conditions. If you can rub it on your skin, that's the goal. And the way that it works is that with the Sorlins, first you have to, rub it on with the cream. It gets inside your cells, and then you shine a blue light on it. Or blue UV range. There are specific light wavelengths that cause Sorlin to make covalent bonds, covalent adducts with neighboring nucleotides. So the actual treatment is called PUVA treatment, Sorlin plus UVA treatment.
Okay. And then this project that you're working on, it sounds like there's quite a connection between the medical field, or a direct application within the medical field of what you're looking at. So maybe just expand, if you don't mind, a little bit about the possible applications of what you're studying are on the medical field.
Mm-hmm. Yeah, so like I hinted before, in long term, once we figure out what these rules are in terms of neighboring nucleotides, we can get a consensus sequence. We can get a stretch of DNA nucleotides where these particular chemicals, and each one probably will have a different sequence, will preferentially interact with the DNA and cause damage. And once we do that, we can get more into the bioinformatics aspect and find out where in the genome these sequences are, if there are specific genes that harbor these sequences, and what do those genes then do? And if we mutate those genes, we mutate those proteins, does this explain some of the side effects that we see? Or if there are environmental toxins, does it explain the illness and the results of those toxins?
Okay, so we were talking before about your research, and you mentioned this idea of trying to figure out exactly where the chemicals or the drugs that you investigate go into the DNA or interact with the DNA. Can you talk a little bit about what you did when you were at Princeton that's along those lines? Because I thought that was really interesting, the idea that you were trying to figure out a very specific sequence in the genome. So go ahead.
Yeah, so this project is an offshoot of my PhD thesis at Princeton. So at Princeton, what I was trying to do was develop basically a form of gene therapy for single point mutations or substitution mutations. And we were using the sickle cell disease allele as our proof of principle. So for those of you who aren't familiar, sickle cell disease comes from a single mutation, a single change in the DNA. One single A is changed to a T in the beta globin gene. And this change is very dramatic. And it was actually the first mutation to be shown to change the protein structure, and the first disease, therefore, to be attributed to a change in the protein structure. So there's a long history with sickle cell disease. And what we were trying to do was basically fix that one little itty bitty change, right? One little change in the genome that should be fixable, one would think. So the lab that I worked in used triplex technology, so making 3-stranded DNA, which is very, 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 very specific targeting that's allowed. If you have so much as one change in the sequence, it's not going to interact, it's not going to work. For those of you who have done PCR and designed PCR primers in any type of lab research, that's making 2-stranded DNA. And you may know that you have a lot of flexibility in terms of what's allowed mismatches in that DNA. That's not the case with triplex. So it's really good for applications like gene therapy, where you want to know that that third strand is only going to go to the location in the genome that you're targeting. And what we were targeting to that point mutation was the chemical sorlin. So sorlin is, like I said, these are flat, planar molecules. They can slide in between the rungs of that DNA ladder. And if we put that sorrelin so it goes right next to that sickle cell allele, that mutant T, and then shine light on it, cause DNA damage, the cell is going to see that and repair it. And DNA damage repair mechanisms are inherently error prone. But what's good is that the most common error it makes in that situation is to change the T to an A, which corrects the sickle cell mutation and actually puts it back to the sequence that we want. And my job in this whole project was to start moving this more into live cells. Can we do this in live cells? So I needed to make some type of a report or a readout for our troubleshooting efforts. So in making that readout, the logistics of putting pieces together, I ended up having to shorten the sequence where that third strand bound by one base pair. And in doing so, what I found is that instead of the sorrelin going right next to the T we wanted, it flipped completely. It went in upside down and backwards. So something was drastically different that would cause that to go in the wrong location, which was not expected. All I did was shorten one base pair. And it wasn't even like I put a different sequence there. It was a duplicated base pair. So big changes for not a big difference.
Yeah,
And
so
that kind of got me thinking, there have to be more specific rules. Really, the only rule that's known is that sorrelin likes to attack pyrimidines, and it likes T's better than C's. But there has to be some type of a neighbor effect.
yeah, it sounds like maybe the entire sequence that surrounds that site is going to play an important role. And
Mm-hmm.
I've studied cancer biology throughout my career, and one of the common ways to teach about mutations and DNA repair is talking about thymine dimers, which is somewhat similar to what you're mentioning. Um, but we know that those thymine dimers do occur more often in certain places in the genome, and that has to do with the surrounding DNA as well. It sounds like the drug interactions or chemical interactions with our DNA maybe
Mm-hmm.
are very similar in that respect, and so that that's really interesting. Especially in those drugs or chemicals where we do see a lot of side effects.
Mm-hmm.
Understanding what those are and why they come about, I think would be a big step forward in the treatment, or
Mm-hmm.
in the use of certain chemicals or drugs for treatment. So
Yeah.
excellent.
So something to remember in my PhD project, we had tethered it and such a short tether to that third strand that it could literally go plus or minus one base pair, and that was it.
Hmm.
And even with that constraint, it managed to go in upside down and backwards. Now, in the medical, PUVA treatment, sorrelin plus UVA, it's not constrained to where it has to be located. It's just free sorrelin that's introduced into the cells. So if one teeny tiny change at a position can create that drastic of a difference of interaction,
Hmm.
what happens when it has a whole genome to play with?
Right. Wow, that's interesting. Well, I hope that the work that you're doing there will lead to some interesting findings for our understanding. Um, is there anything else that you're working on or that you hope to work on in the future?
Yeah, so right now, like I said, this is the active project in the lab. In the future, I don't know where this project is going to go. We may get answers quick, we may get answers slow. I can't predict that. That's the nature of science. Throughout my career, I have managed to pick up a string of projects along the same lines. I saw something weird, but just didn't have time to investigate it. So I have, like I said, a whole string of projects that now potentially have time to look into.
Yeah, that's the thing about science, right? You always find something interesting that you want to look at further, but finding the time to actually do that can sometimes be a bit difficult. Well, here before we wrap up, um, based upon your experiences in the field of science and research, and now your interactions with students here at KCU, what's one piece of advice that you would give to a student, whether they're pursuing a medical degree or a master's degree that's based in research? What's a piece of advice that you think would be beneficial for students to hear?
Mm-hmm. So I think my one piece of advice would be that science is more than just the techniques. So think about your projects, think about them more in terms of what you're interested in, and then how can you use what you've learned and apply it to a completely different situation. So in my experience, I have jumped from lab to lab to lab, from topic to topic to topic. Like I said, in my PhD research, I was looking at third strand, making three-stranded DNA, trying to do gene therapy. I then went to a postdoc that worked on tuberculosis. My second postdoc, I worked on a materials application using the silica shell of diatoms as a solid state platform. For industrial aspects. And, all of those are related by a common thread. They used molecular biology techniques. So you can apply what you know to various fields. Don't constrain yourself to just, thinking, oh, I only know how to do one technique. That's all I can do. That's the only field that I can stay in. Realize that you can apply your knowledge in whatever situation you're in. You just need to take a little creativity and problem solving, and you can find a solution.
Great, great. Yeah, that's great advice. Um, well, Dr. Ford, I really appreciate you taking the time to meet with me today and to really give the KCU community a little bit more insight on where you're at and what you're doing in the lab. Um, I think we all hope that soon we can be in the lab a lot more. It's harder to do this research from home, but um, we hope that we can get into the lab and start moving on some of this stuff. And I know you're looking forward to that as well. Um, and as we do, hopefully this will give some students some background on where your interests are, as well as your colleagues here at KCU in terms of making collaborations and things like that. So it was great talking with you, and thanks again.
You're welcome.
Have a good
Thank
day.
you for
Thank you.
inviting me.
No problem. No problem. Thank you.
All right.